Welcome to this session on the basics of neuroscience. Today, we are going to explore a fascinating and absolutely vital part of human anatomy known as the ventricular system. If you have ever wondered how your delicate brain survives the constant bumps and movements of daily life without getting damaged against your hard skull, you are in the right place. We will uncover the biological plumbing network that keeps your mind safe, floating, and clean. To start, let us build an intuitive understanding of why this system is so important. Imagine carrying a very ripe, delicate peach inside a hard plastic lunchbox. If you shake that box even slightly, the peach will smash against the rigid walls and bruise instantly. Now, imagine filling that lunchbox completely with water so the peach floats perfectly in the center. If you shake it now, the water absorbs the shock and the peach remains perfectly safe. Your brain is exactly like that delicate peach, and your skull is the hard plastic box. The ventricular system is the biological plumbing network that produces and circulates the protective fluid, keeping your brain safely suspended and protected from impacts. Now that we understand the protective role of this fluid, let us look at the physical structures that make it possible. The ventricular system is formally defined as a highly specialized, interconnected series of hollow cavities located deep within the core of the brain. These cavities are lined with specialized cells called ependymal cells. Inside, they contain a crucial structure called the choroid plexus, which acts as a biological filter to produce cerebrospinal fluid, commonly known as CSF. The system consists of four main chambers: two lateral ventricles in the cerebral hemispheres, a third ventricle in the midline, and a fourth ventricle near the brainstem. These chambers are connected by narrow channels, such as the cerebral aqueduct, allowing the fluid to flow continuously. Having discussed the anatomy, let us now visualize these structures inside the human head. On your screen is a lateral or side view of the brain. The light gray outline represents the brain tissue, while the bright blue structures deep inside represent the ventricular system. Notice the large C-shaped lateral ventricles at the top. Fluid flows from these down into the central third ventricle. From there, it travels through that narrow downward channel you see, labeled as the cerebral aqueduct, finally reaching the fourth ventricle near the bottom. This diagram clearly shows how deeply embedded and interconnected this plumbing network is within our central nervous system. Seeing how the system is laid out brings us to a critical question. Why is this constant fluid production so essential for our survival? Without the ventricular system, our brains would literally crush themselves under their own weight. A typical human brain weighs about 1,400 grams. However, because it floats in the cerebrospinal fluid produced by the ventricles, its effective weight is reduced to just about 50 grams. This incredible buoyancy prevents the delicate neural tissue and blood vessels at the base of the brain from being compressed. Furthermore, this system acts as a crucial waste disposal mechanism, flushing out toxic metabolic byproducts that accumulate while we are awake. If this system gets blocked, it leads to a severe condition called hydrocephalus. To truly appreciate how dynamic this waste disposal and cushioning system is, we must look at the mathematics of fluid management. The formula on screen calculates the turnover time of cerebrospinal fluid. Conceptually, this tells us how long it takes for the brain to completely replace its protective cushion. In this equation, T represents the turnover time, usually measured in days. V represents the total volume of cerebrospinal fluid present in the system at any given moment, measured in milliliters. Finally, R represents the rate of fluid production by the choroid plexus, measured in milliliters per day. Physically, if the production rate R increases while the volume V stays the same, the turnover time T decreases, meaning the fluid is refreshing much faster. Now that we have our formula, let us apply it to a healthy adult human to see this system in action. We are given that the total volume of cerebrospinal fluid, V, is approximately 150 milliliters. The choroid plexus produces new fluid at a constant rate, R, of 500 milliliters per day. We are asked to calculate the turnover time, T. We choose our turnover formula because it directly relates volume and rate to time. By substituting our given values, we divide 150 by 500. This calculation yields a final answer of 0.3 days. Physically, this means it takes about 1/3 of a day for the brain to completely replace all of its fluid. Having found that it takes a fraction of a day to replace the fluid, this naturally leads us to ask, how many full replacements happen in a single 24-hour period? To find this, we use a simple reciprocal relationship. The formula is N equals 1 divided by T. Here, N represents the number of complete fluid turnovers per day, which is a measure of the system's efficiency. T is the turnover time in days that we just discussed. Conceptually, taking the reciprocal of the time required for one cycle gives us the total number of cycles that can fit into one standard day. If the turnover time T becomes smaller, the number of daily turnovers N will physically increase, indicating a highly active system. Let us continue with our previous example to find exactly how many times the fluid is replaced. We know from our last calculation that the turnover time, T, is 0.3 days. We are asked to find N, the number of turnovers per day. We use the reciprocal formula because it converts the duration of one cycle into a daily frequency. We substitute 0.3 into the denominator. 1 divided by 0.3 gives us approximately 3.33. Physically, this final answer means your ventricular system completely flushes and replaces the entire volume of cerebrospinal fluid more than 3 times every single day. It is a highly dynamic system, constantly working to keep your brain clean. We have seen how beautifully this system works when healthy. But what happens when things go wrong? Imagine a patient develops a blockage in their cerebral aqueduct. This leads to a dangerous condition known as obstructive hydrocephalus. The critical issue here is that the choroid plexus does not know there is a blockage. It continues to produce fluid at its normal, constant rate. However, because the exit pathway is blocked, the fluid can only drain at a severely reduced rate. Consequently, excess fluid becomes trapped inside the brain, causing the ventricles to swell rapidly. This swelling creates immense internal pressure, which can crush the delicate brain tissue against the rigid skull, making it a severe medical emergency. To mathematically understand the severity of this blockage, we first need to determine how fast the fluid is building up. This brings us to the net rate of fluid accumulation formula. Conceptually, this is a simple balance equation: what goes in minus what comes out. In this formula, R net represents the net rate at which fluid is being trapped, measured in milliliters per hour. R in is the rate of fluid production by the choroid plexus, and R out is the severely reduced rate of fluid drainage. If the drainage rate, R out, decreases due to a blockage, while production, R in, remains constant, the net accumulation rate, R net, physically increases. A positive net rate is the mathematical root cause of the swelling. Let us apply this net rate formula to a specific patient scenario. We are given that the choroid plexus continues to produce fluid, Rn, at a rate of 20 milliliters per hour. Due to the blockage, the fluid can only drain, R out, at a reduced rate of 5 milliliters per hour. We are asked to find the net rate of accumulation. We use our balance formula to find the difference between production and drainage. By substituting 20 for R in and 5 for R out, we subtract 5 from 20. The final answer is 15 milliliters per hour. Physically, this means that every single hour, 15 milliliters of excess fluid is being permanently trapped inside the patient's brain. Now that we know how fast the fluid is trapping, we need a formula to predict exactly how much the ventricles will swell over a specific period. This is the volume accumulation formula. Conceptually, it states that the final size is simply the starting size, plus whatever new fluid has been added over time. Here, V final is the swollen volume of the ventricles. V initial is the normal starting volume. R net is the accumulation rate we just learned to calculate, and T is the time elapsed in hours. Physically, as the time elapsed T increases, the term in the parentheses grows larger, causing the final volume, V final, to increase dangerously. This equation models the rapid progression of the medical emergency. Let us finish our patient scenario by calculating the total swelling. We're given an initial ventricular volume of 50 milliliters. From our previous step, we know the net accumulation rate is 15 milliliters per hour. We are asked to find the final volume after a time elapsed of 4 hours. We use the volume accumulation formula to project the total growth. First, we substitute our values into the equation. We multiply the net rate of 15 by the 4 hours elapsed, which gives us 60 milliliters of newly trapped fluid. We then add this 60 to our initial 50. The final answer is 110 milliliters. Physically, this shows that in just 4 hours, the fluid volume has more than doubled, illustrating why immediate medical intervention is required. We have covered a lot of ground today, so let us review the key takeaways. First, the ventricular system is the brain's vital internal plumbing network, providing essential buoyancy and physical protection. Second, by continuously producing and circulating cerebrospinal fluid, it acts as a dynamic waste clearance system. Third, we saw how mathematical formulas allow us to calculate fluid turnover and predict the severe, rapid consequences of blockages like hydrocephalus. You now have a complete, intuitive, and mathematical grasp of how this beautiful system keeps your mind afloat and safe. Great job working through these concepts.